Alright everyone, welcome back for part 2 of our lab presentation on the reproductive system. In the last lab recording, we focused specifically on the anatomy of both the male reproductive system and the female reproductive system. But in this lab recording, we're going to specifically look at the physiology of both reproductive systems. The reproductive systems are quite unique from our other 10 body systems. When merging the physiology of both the male and female reproductive systems, we get the phenomenon of reproduction. This lab recording specifically will focus on how each of these reproductive systems produce gametes, and we will also briefly touch upon the concepts about what happens when a male gamete meets a female gamete. So we must first define our terms if we're going to understand the reproductive system and its mechanisms. The first term that we're going to discuss is known as gametogenesis. Gametes are sex cells, and genesis means the formation or production of. So gametogenesis refers to the production of reproductive cells. This is a process that happens in both males and in females. However, in males it's called something different than what it's called in females. In females, we call this the process of oogenesis, which is the production of the female reproductive cell known as the oocyte. Oogenesis is gametogenesis, but specifically gametogenesis in the female. Spermatogenesis is our term for gametogenesis that takes place in males. The process of spermatogenesis will produce spermatids, which are immature sperm cells. However, in males, spermatogenesis is not the only process that falls under the blanket term of gametogenesis. We also have a process known as spermiogenesis, which takes these spermatids formed from spermatogenesis and turns them into mature sperm cells, which are known as spermatozoa. This is more of a modification process, but it still falls under the process of gametogenesis. Another word for gametogenesis is meiosis. If you can think back to Anatomy and Physiology 1, we would have discussed mitosis and cell division. This was the process that consisted of prophase, metaphase, anaphase, and telophase, and most of the cells in our bodies will actually divide through the process of mitosis. But meiosis, or meiosis on the other hand, is a type of cell division that's specific to gametes. Let's take a look at these differences. Mitosis is a process that produces what are known as diploid cells. Diploid cells will contain two sets of chromosomes, one from mom and one from dad. But we see that meiosis is actually going to produce a different type of cell known as a haploid cell. Haploid cells, by definition, have half of the chromosomal material as diploid cells do. The whole purpose of producing haploid cells is to produce a cell with 23 chromosomes that can meet with another cell that contains 23 chromosomes from another person. When those two cells come together, we mix that genetic information and we get a new set of DNA. In humans, diploid cells contain 46 chromosomes and haploid cells contain 23 chromosomes. Again, thinking about mitosis, we took one parent cell and divided it into two daughter cells. But those two daughter cells were exactly identical to the parent cell, and each daughter cell contained 46 chromosomes. The whole purpose of mitosis was to facilitate growth and repair, as the cells in our bodies are constantly being damaged and stressed. The purpose of meiosis, however, is not to produce more cells to replace or repair tissues. The sole goal of meiosis is to produce gametes that will be used for reproduction. Gametes are in fact the only cell in the entire body that will divide by meiosis. We can see here that the goal of mitosis is to produce two identical diploid cells, and the goal of meiosis is to produce 4 non-identical haploid cells. Let's take a look at this process in the diagram on the right. Starting with meiosis, gametes are going to develop in the reproductive systems of both males and females. Meiosis produces haploid cells that contain 23 chromosomes each. When the sperm cell penetrates the oocyte, that's a process known as fertilization. And from here on, we now have 46 chromosomes that can be found in what's known as the diploid zygote. The word zygote means a newly fertilized egg cell. So once two cells with 23 chromosomes each come together, that gives us our first diploid cell. This cell is going to continually undergo the process of mitosis for growth, repair, and development. Eventually, after many divisions, our diploid cell is going to become what we know as an organism. And as the reproductive systems developed, they will be honed to produce haploid cells. This again will happen through the process of meiosis, and we will repeat this cycle over and over. Meiosis can also be referred to as reduction division, because we're not only dividing our cells once, we're actually going to divide each cell twice, which is how we get 23 chromosomes. Mitosis and meiosis are side by side here for comparison. With mitosis, the parent cell is going to have a total of 46 chromosomes. 23 of those chromosomes came from mom, and 23 of those chromosomes came from dad. We're only depicting one pair of chromosomes in this parent cell, but there will be 23 pairs of chromosomes, as both mom and dad provided one set. Through the S phase of interphase, we're going to replicate each of these chromosomes, and we're going to package them up during prophase so that they can eventually be divided into two of our daughter cells. From here, metaphase, anaphase, and telophase will take place, and we'll have two daughter cells that are identical to the parent cell that produced them. With meiosis, on the other hand, there's going to be some differences. We'll have the diploid parent cell, and we're also going to have the replication of the DNA. But here with meiosis, we actually run through mitosis twice. We will first have prophase 1, metaphase 1, anaphase 1, and telophase 1, and then we will jump directly into prophase 2, metaphase 2, anaphase 2, and telophase 2. To understand this, you can see that prophase 1, metaphase 1, anaphase 1, and telophase 1 are essentially the first round of mitosis. There are some other events taking place in here that will not result in identical diploid cells. However, after telophase 1, we do have diploid cells that would have 46 chromosomes each. From here, instead of jumping into interphase like we would do with mitosis, these two cells here are eventually going to divide once more through the process of meiosis 2, and that's going to separate these chromosomes one more time to leave us with 23 chromosomes each. From here, these will be viable gametes that can go and meet with another gamete that is also considered haploid, and together that will produce a new diploid organism that is unique in its own right. Both male and female gametes will undergo the process of meiosis, but the results in each system will be slightly different. Spermatogenesis and spermiogenesis will produce 4 viable haploid gametes, but the process of oogenesis is only going to produce 1 viable oocyte. Let's first discuss the process of spermatogenesis and spermiogenesis, and then we'll make our way into the process of oogenesis. So as we discuss the anatomy of the male reproductive system in our last lab recording, we will note that the seminiferous tubules are going to be the location where spermatogenesis is going to occur. This process starts with diploid spermatogonia, which are the stem cell for the male gamete. Through the process of meiosis, spermatogonia are going to develop into what are known as haploid spermatids. Remember that meiosis goes through prophase, metaphase, anaphase, and telophase twice, and therefore we're going to have the transition from diploid spermatogonia into haploid spermatids. Spermatids, however, are not fully developed and would not be able to make their way out of the male body, so they have to undergo a second process known as spermiogenesis, which refers to the modification of spermatids into spermatozoa. Spermatozoa will contain the inflamous flagella, and these are in fact the only human cells that have been discovered to have this organelle. The spermatogonia will be hanging out on the outskirts of each seminiferous tubule, and as they further develop into spermatids, they're going to make their way towards the lumen of these seminiferous tubules. After spermatozoa have been formed, they will be released into the lumen and be carried out to be stored in the epididymis for later release. We want to be very careful and understand the difference between spermatogenesis and spermiogenesis. Spermiogenesis refers to the modification of spermatids into the spermatozoa. During this process, spermatids will shed most of their cytoplasm and also develop what is known as an acrosome. That's the little cap on the head of the sperm cell. Additionally, the flagella will form, and that gives the sperm cell its ability to propel itself forward. You can see the head of the sperm cell will contain the nucleus, which will have the 23 chromosomes that make this cell haploid. Just a little extra information on sperm cells. Spermatozoa in particular will have 3 major parts. They'll have a head, a middle piece, as well as a tail. I've already mentioned that the head contains the nucleus, which will also contain the genetic information, and it will have an acrosomal cap as well that sits on the nucleus. This acrosome contains digestive enzymes that will help the sperm cell penetrate the oocyte in the female reproductive system. The tail is also known as the flagella, and we know that flagella are used to propel these cells forwards. But the middle piece actually contains lots and lots of mitochondria that will produce energy for the sperm cell and allow them to facilitate movement of the flagellum. We have seen this slide several times before. We are looking at the seminiferous tubules. You can see one seminiferous tubule shown right here, but there are many of these that can be found throughout the testes. Specifically, closer to the outer margin of our seminiferous tubule, we will find those diploid spermatogonia. These cells will be found around the outskirts of the seminiferous tubule, and as we move closer to the lumen, we're going to start seeing spermatids. Hanging on the edge, or perhaps in the lumen, we can actually see the mature spermatozoa that are ready to go for fertilization. In case we don't remember from the endocrine presentation, the interstitial cells of Leydig will also be found in the testicles. These are producing testosterone. Testosterone is a very important hormone that will be used to facilitate the process of spermatogenesis. Since spermatogenesis will be stimulated by testosterone, we need to go back to the endocrine system to discuss testosterone's release from the Leydig cells. So the hormonal control of spermatogenesis is truly facilitated by the endocrine system. We can hopefully remember that the hypothalamus is the major regulator in our brain of the endocrine system, and this particular part of the brain releasing hormones that will stimulate other hormones to be released from the anterior pituitary gland. The releasing hormone of interest to the reproductive system is gonadotropin-releasing hormone, or GnRH. When gonadotropin-releasing hormone from the hypothalamus makes its way down to the anterior pituitary gland, that's going to stimulate the release of follicle-stimulating hormone and luteinizing hormone. FSH and LH will eventually enter the bloodstream and travel down to the testicles. Each of these two hormones will work together to stimulate the process of spermatogenesis, and LH specifically will act on our Leydig cells to tell them to release testosterone. Both FSH and LH will stimulate the spermatogonium. This causes them to increase their activity and start producing more spermatids. All the while, LH is also going out to our interstitial cells of Leydig and telling them to release testosterone. And then testosterone will further feed back on the seminiferous tubules to further stimulate the process of spermatogenesis. Spermatogenesis is nearly a lifelong process. It begins at puberty but continues throughout life. Oogenesis is going to work a little bit differently. This process actually begins at birth, arrests for a little while, and restarts at puberty. But I also mentioned a little bit earlier that oogenesis does not produce 4 viable haploid cells. Normally, only one oocyte is produced each month by the process of oogenesis. And this process has several checkpoints that will be required to finish the development of the oocytes. Through the process of oogenesis, only one viable haploid cell is produced. So you might be wondering where the other 3 haploid cells are. Instead of producing other viable cells, the process of oogenesis results in what are known as polar bodies, which are uneven distributions of the cytoplasm of our divided diploid oogonium. We will still have a meiosis I and a meiosis II, but certain events must occur in order to complete the entire process. Meiosis II will only take place if fertilization from a sperm cell has occurred. If it does not, the oocyte is going to eventually break down, and this is the basis of the uterine cycle. So I mentioned before that the process of oogenesis actually starts occurring before birth. Instead of the spermatogonium, we will have the diploid oogonium, and this is going to produce what's known as the primary oocyte that will stop in prophase I when the child is born. Once puberty begins, we're going to continue the process of oogenesis in the female. However, this is going to facilitate much differently than it did in the male. Sperm cells are constantly being produced in the male, but in the female specifically, the uterine cycle will regulate the development of a single primary oocyte each month. And so it is every month that one oocyte will go through the process of meiosis. We start with the primary oocyte that's in prophase I. And as it goes through metaphase I, anaphase I, and telophase I, it's eventually going to divide and form what's known as a secondary oocyte. This would then be considered a haploid cell. The secondary oocyte will be ready for fertilization by the sperm cell, And once the sperm cell has penetrated, we're going to call this a zygote. Now, it would seem that there's an uneven distribution after the process of meiosis I. And this is in fact correct. Instead of producing two secondary oocytes, we have an uneven division of cytoplasm that produces the first polar body. This polar body may go on to divide again, which will form the first and second polar body. If the secondary oocyte itself divides once more, that's going to form the third polar body. But despite having all these products, the secondary oocyte is the only viable gamete that's produced through meiosis. In order to nourish and facilitate the development of oocytes, each oocyte is going to be found in a structure known as an ovarian follicle. Ovarian follicles are circular structures that wrap around the oocyte and provide nourishment to facilitate the growth and development of this cell. At puberty, the oocyte is going to be wrapped up in a primary follicle, which will develop into a secondary follicle, which will further develop into the mature Graafian follicle. This mature Graafian follicle will be ready to release the secondary oocyte out of the ovaries and into the fallopian tubes, where fertilization can take place. We have also seen this picture several times before, but now we're going to focus on all of the structures that are labeled on this slide. So we'll first see the oocyte shown right here. The oocyte will have its own nucleus, and it's going to be covered by this layer known as the zona pellucida. The zona pellucida will stick with the oocyte even after it's been released from the ovarian follicle. And it is this layer that the sperm cells will have to penetrate with the acrosomes that are carried on their heads. Covering up the zona pellucida, we'll find the corona radiata shown right here. These cells will also follow the oocyte once it has been ovulated. This structure right here is a large fluid-filled space known as the antrum. And wrapping around the entire ovarian follicle are the follicular cells, which were in fact the cells that released estrogen. The Leydig cells in males release testosterone, and the follicular cells in females will produce estrogen. This slide nicely shows us that the ovarian follicles are developing at different times. Each month, one ovarian follicle is going to reach maturity and release the oocyte from the ovary. The release of an egg cell from the ovary is known as ovulation. Once the egg cell has been ovulated, the fimbriae of the fallopian tubes are going to waft this cell over to enter the fallopian tubes, where this cell can be fertilized in the event that sperm cells are present. While the oocyte is making its way through the fallopian tube and into the uterus, the follicular cells are going to undergo a transition into what's known as the corpus luteum. The corpus luteum can be shown right here, and its major role is to produce progesterone that will stimulate the growth of the endometrial layer of the uterus. The hormonal control of oogenesis is also facilitated by the endocrine system. Just as we saw in the male, in the female, the hypothalamus is going to release gonadotropin-releasing hormone. This is going to feed back on the anterior pituitary gland to release follicle-stimulating hormone and luteinizing hormone. Instead of stimulating spermatogenesis, follicle-stimulating hormone gets its name as it actually stimulates the development of the primary follicle. As FSH is focusing on this development, luteinizing hormone will stimulate the ovary to ovulate the egg cell. We saw a couple slides ago that the follicular cells are going to be producing estrogen. Estrogen will be used to develop the inner lining of the uterus known as the endometrium. This is going to be necessary for the implantation of the blastocyst into the wall of the uterus. But I also mentioned a couple slides ago that after ovulation, the follicular cells become a structure known as the corpus luteum. And once the corpus luteum is present, it's no longer going to be producing estrogen. In fact, it's going to switch and start releasing the hormone progesterone. And the action of progesterone is simply to also feed back on the endometrium to continue its growth and development. This is a good slide for us to look at, as it will show us the timing of these hormonal events in tandem with ovulation. The first thing that we need to note is that on average the uterine cycle lasts 28 days. Certain individuals might slightly deviate from this number, but 28 days is the average. Based upon the uterine cycle being this long, 14 days is going to be halfway through the cycle, which is actually in fact when the release of follicle-stimulating hormone and the release of luteinizing hormone are going to spike. Follicle-stimulating hormone is stimulating the development of the primary follicle, while luteinizing hormone is stimulating the ovulation of the oocyte from the ovary. And in fact, here at day 14, when LH is spiking, we can actually see that that's going to be the time when ovulation takes place. Once ovulation has occurred, the follicular cells are going to become the corpus luteum, and this structure is going to start releasing progesterone. And in fact, we can see that there's virtually no progesterone release until ovulation has taken place and the corpus luteum has been formed. Once the corpus luteum is present, progesterone will start being released to continue the growth of the endometrium. Estrogen had started the growth of the endometrium, and this was being secreted by the follicular cells. That's why we'll have a bit of estrogen release shown right here. As FSH is stimulating follicular development, the more FSH that's released, the more estrogen that's going to be released. And that's why we see the spike of FSH and estrogen at virtually the same time. The last thing that we want to notice is that the thickness of the endometrium is increasing throughout the uterine cycle. Since estrogen is starting the growth of the endometrium and progesterone is continuing the growth of the endometrium, you can see that this lining will get thicker and thicker as the days of the uterine cycle progress. But at day 28, if no fertilization has taken place, the endometrium will slough off and the uterine cycle will begin again. This cycle will repeat if no fertilization takes place. However, in the event that sperm cells are present and fertilization does in fact take place, that's going to lead to the formation of a new diploid zygote that will continue to divide until it has enough cells to be referred to as the blastocyst. The blastocyst will eventually implant in the wall of the endometrium, and certain cells contained within the blastocyst, known as the trophoblast, are going to release another hormone known as HCG. HCG stands for human chorionic gonadotropin. And this hormone is going to eventually feed back on the corpus luteum to maintain it and tell it to continue to release progesterone. In response to this, progesterone will then feed back to continue the growth of the endometrium that will continue to nourish the blastocyst. This process is described on this slide. So inside of the blastocyst, there's a group of cells known as the trophoblast. The trophoblast cells are going to form in the blastocyst. And once the blastocyst has implanted into the wall of the uterus in the endometrium, the trophoblast cells are going to start to release human chorionic gonadotropin. HCG will feed back on the corpus luteum and maintain its function of releasing progesterone. Progesterone continued endometrial growth, so we're going to see the maintenance of the endometrium and also the prevention of further ovulation taking place. Once the trophoblast cells have started releasing HCG, this is a fairly good indicator of pregnancy, and therefore it is the hormone that's detected by many different pregnancy tests. The last concept that I'm going to mention in regards to reproductive physiology is the importance of body fat in women. A normal body fat percentage is between 22 and 25%. Athletes may have a little bit less than this, but this is still quite a good amount. Body fat will actually be found producing some estrogen. And so in the event that a woman has very low body fat, this can lead to reduced fertility and overall loss of bone mass. Adipose tissue is often given a bad rap, but this is just one example to show actually how necessary adipose tissue is for the homeostasis of the body and the homeostasis of the female reproductive system. This will conclude our unit on the reproductive systems. I will see you all again for our final lab recording that will cover surface anatomy and heredity.